So let's talk about chromosomes just for a moment. So I showed you the structure of chromosomes just a moment ago. Tightly coiled DNA. What I want you to do, we're going to draw a portion of a karyotype. Okay. So here's our karyotype. Okay. And like I mentioned, you have 23 pairs. We're just going to do the first 4, bless you, and then we're going to do the 6 chromosomes, bless you. So the way a karyotype is actually done is that when they're isolated, the chromosomes are isolated, they have what's known as a staining pattern. They have bands on them. If you notice, they have like little lines on them, and there's a way to do that that helps them identify each type of chromosome. Because what's unique about each type of chromosome is that the 2 copies, say, of chromosome 1, they contain the same set of genes. Let me say that again. All right, the 2 copies of chromosome 1, they contain the same set of genes because it's the same portion of the human genome, or the collection of DNA that humans have, right? So I'm going to indicate with red and green maternal and paternal chromosomes. Okay, so maternal chromosomes are going to be in green, and paternal are going to be in red. Okay? So chromosome 1, I've got 1 from my dad and 1 from my mom. And so they match them together with their banding pattern, so it looks like when they stain them, it's like each chromosome has like a fingerprint, and that has to do with a chemical that they expose them to. But what's also cool is that chromosomes 1 through 22 are arranged in descending size. Right, chromosome 1 is the largest. Chromosome 22 is the smallest. Right? So it's not a huge jump between 1 and 2, but just so you can see, it looks something like that. And then a little bit smaller. And a little bit smaller. This is probably not the most accurate, but just so you see the idea that it's descending in size. And each of these would have their own unique banding pattern. There's actually this really cool biotechnology technique called FISH, and it stands for fluorescence in situ hybridization, and they actually put these like glowing markers onto the chromosomes, and it looks like this really pretty photo. I'll show you a picture of that tomorrow, but one of the ways they can identify chromosomes. Alright, so like I mentioned, 1 through 22, these are autosomes. And then you have the 23rd pair, which is your sex chromosome. So in this case, from the dad, let's say a Y was contributed, and from the mom, let's say an X chromosome was contributed. So this would be a biological male, XY. Okay. What I want to also show you is that if we were to zoom in a little bit here, to what's going on with these chromosomes. There's a couple of terms that honestly are kind of confusing because they all sound the same, but I want to just go over them real quick because I'm going to use them a lot. So, we've got maternal and paternal chromosome 1. This is prior to replication. When I replicate the chromosomes, they start looking like this. So I've replicated both my maternal and paternal chromosome 1. And they're connected, this point right here, they're connected by something called a centromere. All right, that's what connects the two replicated chromosomes. Okay. Now, what they use to describe is that if you're talking about the same chromosome that's been replicated, we call these two strands sister chromatids. Sister chromatids. Right? Now, when you refer to the pair of maternal and paternal chromosomes, we call these homologous chromosomes. And that just simply means that they represent the same portion of the human genome. Their genes are homologous. Okay. Now, remember when I showed you that photo where you had the DNA kind of unraveled like that, right? Because DNA is coiled. When DNA is coiled around histones to make a chromosome, the word that they use is called chromatin. Again, this is where all the terms kind of mix together. We got chromatin, chromatids, and chromosomes. Okay. And lastly, when you refer to the replicated homologous chromosomes, all four, we call that a tetrad. A tetrad is the replicated version of the homologous chromosomes. Okay. All right. Now, within these homologous chromosomes, I mentioned that they have the same set of genes, right? And so what we do is we refer to a portion of the sequence of a chromosome. Like if I'm looking at that particular part of the DNA, we call this a locus. Right? A locus is a portion of the chromosome, a particular sequence. Doesn't necessarily have to be a gene. Sometimes you're just looking at a random sequence. But what we see is that on the homologous chromosomes, the locus or the loci are about the same position. All right, so on that corresponding part, and that's why they use the banding pattern so they can kind of see which direction the chromosomes are in. The locus will be the same for that sequence. Okay. Any questions so far? All right. So, those are chromosomes. Now let's talk about mitosis. So, mitosis... Oh, we got space down here. Mitosis has probably about three major roles, right? So mitosis is a type of cell division that's mainly used for one, growth of an organism, something that, as a developmental biologist, that blew me away, that got me into the field of developmental biology, is that you start as one zygote, one diploid cell, and your body contains approximately 35 trillion cells. Not only that, but as they divide, they become specific for different functions. And we're going to talk about that in the next unit. It just blows me away. But growth of an organism. Number 2, repair or replacing. I think Alexei was mentioning this yesterday. Dead or lost cells. Okay. And then the third, for some organisms, mitosis is used for asexual reproduction. For example, I mentioned this yesterday, bacteria use a form, kind of a similar form of mitosis in something called binary fission. But plants will use mitosis as well for asexual reproduction. All right. Those are the roles. Let's talk about the cell cycle. So, Unit 4 mentioned this yesterday, but the cell cycle, we're going to use lots of space for this. So the cell cycle it's always so difficult to draw a circle. Did anybody grow up watching SpongeBob? There's the episode where SpongeBob is taking an art class, and Squidward is asking him to draw a perfect circle, and he draws this amazing face, and then he erases it, and it's like a perfect circle. And Squidward's like, "How did you do that?" He's like, "Well, first I draw this face, and then I erase this, and then I erase this, and then voila, perfect circle." And then Squidward's like, "No, just draw a circle," and then he draws some thing like this, where it's all lopsided. All right. I don't know why, but I've been thinking a lot about SpongeBob references lately. Sometimes people think they're funny, sometimes not. All right. So we have our cell cycle. Looks like this. We have G1, S, and G2, which compose what's known as interphase. So from here all the way to here, we have interphase. And the reason we use a pie chart looking thing is to make the point that most of the time a cell spends their life in a interphase, either in one of these three. The other portion is referring to the M phase, mitotic phase. And so you have mitosis, and you also have at the end cytokinesis, which is where after mitosis has occurred, the cells actually split into two. All right. So, within this, let's say a cell enters here to this cycle, so they've already divided and they go into G1. G1, all we need to know here is it's typically just growth. The cell is growing in size. Okay. Sometimes at this phase, most cells in our body actually do this for at least a period, is that they'll kind of grow for a little bit, but then they'll enter something called G0 or G0, and that's just, it's not really growing anymore, it's just there. right? So growth kind of stops and it's just functioning. Now, sometimes they'll re-enter G1 and they'll go through interphase for the purposes of replication. At the end of G1, we'll talk about a little bit more in depth, what's known as the G1 checkpoint. Okay. Now, during the S phase, primarily what's happening here is DNA replication, and we're going to talk a lot about DNA replication in Unit 6 and how it replicates semi-conservatively. But that's where DNA, right, this replication of the DNA is occurring. So we got the S phase. And then G2, this is where we tend to have more growth and prep for mitosis. There's some kind of final checklist items that the cell has to make sure that it has before it goes on to the M phase. And at this point we have something known as the G2 checkpoint. Okay. Now at the end of mitosis, right about here, we have the M checkpoint, the M phase checkpoint. Okay. And that's just to make sure that if anything went wrong during mitosis, like the separation of the chromosomes, that it can instead induce apoptosis or repair the damage. Okay. So something that often trips people up when you think about this is the whole thing about CDKs, cyclins, and how they're related to MPFs, all these acronyms that were mentioned last semester. But I want to clarify a couple of these. So what happens here is that if a cell enters right here at the beginning of G1, it will go into G0 unless it receives a growth signal. So the way that a cell is able to pass the checkpoint is if a growth signal is received, right? And this can be like a growth factor. It can be a signal that's actually sent out from a cell nearby, or it can be a cell signaling from far away, right, as in paracrine signaling or endocrine signaling, right? But if that growth signal is received on that cell's receptors, they're like, all right, it's time to move on to DNA replication, right? And that's because DNA replication, it takes a lot of energy, a lot of resources, so there's no reason to go on to S phase if you're not going to ultimately replicate, right? All right. Now what happens here is that they go into the S phase, and it's at this time, about halfway through, that part of that growth signal in DNA replication occurring is that a protein called a cyclin starts to be produced. All right, remember, one of the results of cell signaling is gene expression, right? Cyclin starts being produced. All right, it's this protein. I'm going to use like this, and Mr. Anderson talked about this a little bit. But that protein starts to accumulate inside of the cell, so that by this point you have lots of cyclin entering the G2 portion, right?